Hello, good evening all of you. Welcome to our session. Hope all of you are doing fine. And is there any doubt regarding the topic we were discussing last time? We just started the chapter excretion, that is excretory products and their elimination. In that connection, I told you what are the structures, excretory structures, organs which are present in different animals. I was telling you that in cephalochordata, we are having nephridia with flame cells. Okay. In echinodermata, actually there is no excretory organ, whereas in cephalochordata, in which amphioxus is the classical example, we are having nephridia with flame cells. So, flame cells are the excretory structures in platyhelminths. Let's say in urochordata, in which acidia, herdmania, tunicates are coming. Acidia is a tunicate. Tunicates, you are having neural gland. In hemichordata, you are having an organ called proboscis organ. Which is opening to the proboscis region, so it's called proboscis gland. So we are having so many different types of excretory organs in different types of animals. Remember, animals are excreting this nitrogen based material either in the form of ammonia, that is monotelism, or in the form of urea, ureotelism, or in the form of uric acid, uricotelism. Is that point clear? Anybody having any doubt, Makle? Please respond. Just a minute.
Good evening, all of you.
Open ചെയ്തിട്ടില്ല. ഇത് ശരി, NCERT വരണ്ടേ? So welcome back. Now we pass on to the study of our excretory system. Our excretory system include organs like kidneys, a pair of their ducts called ureters, a urinary bladder, and the duct of the urinary bladder called urethra that opens out by means of the urethral meatus. So these are the structures, organs present in our excretory system. A pair of kidneys, a pair of ureters, a single urinary bladder, and a urethra. Okay. Remember, we are all ureotelic. We send out nitrogen fixed material in the form of a concentrated solution that contains urea. That is why we are all ureotelic. So, our excretory organs are in all vertebrates, it is kidneys. All vertebrates are having kidneys. Frog is having kidneys, we are also having kidneys. Mammals are also having kidneys. Okay. So, kidney is the excretory organ of vertebrates. And each kidney, in our case, is a reddish brown structure, located in the abdomen. What is the correct situation or correct position? Between the last thoracic. Last thoracic means there are 12 thoracic vertebrae. So last thoracic is 12th thoracic. So if you please mark it. Last thoracic is 12th thoracic and third lumbar. Lumbar means vertebrae in the abdomen. So in the thorax you are having 12. So between the last thoracic and the third lumbar vertebra. In between these two vertebrae, on either side of our vertebral column, you are having a pair of kidneys which are all bean-shaped structures having a convexity on the outside and concavity on the inside. They are all connected to the abdominal wall by means of ligaments. That's how they are all kept in position. Kidneys are held in position by ligaments, okay, close to the inner wall of the abdominal cavity. Adult kidneys are weighing about 120 to 170 grams, and in males it is a bit larger than that of the females, having a size of 10 to 12 centimeter in length, 5 to 7 centimeter in width, and thickness is just 2 to 3 centimeters. That is the size, correct size. Remember, weighing about 120 to 170. Females are having only up to 150 grams. Males may be having 170 grams. Okay. Inner side is concave, and towards the center of this inner concave surface, there is a notch. That notch is called hilum. It is called hilum. Okay, this hilum is the point through which the duct of the kidney comes out. Duct of the kidney is called ureter. Okay, the ureter is coming out. So this is the hilum. It is also called hilus. And blood vessel is entering into the, remember, blood is bringing the waste materials. That blood vessel is called renal artery that supply blood to the kidneys. Renal artery arises from the dorsal aorta, which is the main blood vessel. Dorsal aorta. This dorsal aorta gives a branch called renal artery, which enters into the kidney through the hilum, and after entering it branches. We will be coming to that part. Similarly, it is through the same hilum or hilus that blood is taken out. After purification, after filtering out the waste material, blood is taken out through the renal vein, and the renal vein joins inferior vena cava. In circulation, we studied two superior vena cava and one inferior vena cava. Inferior vena cava brings blood from the lower part of the body to the heart. So, renal veins are opening to the inferior vena cava and taking blood away after filtration. Okay. Each kidney is having a tough outer capsular covering made up of connective tissues. This is the capsule, renal capsule, tough capsular covering made up of fibrous connective tissue, within which you are having the tissues of the kidney divisible into two regions. What are they? Namely, an outer cortex and a middle or deep medulla. Okay. Present in this medulla, you are having a few number of conical masses which are in the form of pyramids. So they are called renal pyramids. They are in the medulla. And see, the ureter is taking its origin. This is the ureter duct. It is opening in the form of a wide funnel-like mouth into the kidney. This wide funnel-like structure is called the pelvis. Okay. Renal pelvis. Renal pelvis will be the site to which these pyramids are all focusing. Okay. The pelvis is extending. It is extending. See, this pelvis is extending even to the base of the pyramids. These extensions are called calyx. Calyx is singular, calyx is plural. We are having calyx. Calyx are extensions of the pyramids, in between the pyramids, and in between the pyramids, the cortex is extending. This is the cortex. This cortex is extending, growing in between two pyramids. This extension is called these extensions are in the form of thick structures, which are all in the form of columns. These columns are called columns of Bertini or renal columns. Renal columns, they are called columns of Bertini. These are the pyramids, and in between the two neighboring pyramids, the cortex is extending, growing in the form of renal columns called column of Bertini. So this is the gross structure, general structure of a single kidney. Is that clear? Now, each kidney is made up of a very large number of very small units called nephrons. Study of kidney and associated structures called nephrology. After your MD, you can go for super specialty in nephrology. Okay. So, study of kidneys and associated structures is called nephrology. So, nephrons are the units by which each kidney is made up of. There will be about 1,000,000 nephrons in each kidney. Though we are having two kidneys, one kidney in perfect working condition is more than sufficient for our normal activity. That is why you can donate kidneys. Every now and then we hear about kidney transplantation. Kidney donors are needed. We want a kidney. Who will donate to us? Like that. You will be having appeals appearing in media. So one kidney is sufficient, because each kidney itself is having more than 1,000,000 such nephrons. What is a nephron? Nephron is the structural and functional unit of a kidney. That is the basic unit by which kidney is made up of. That is why it is called structural unit. Kidney is also the functional unit, meaning all the functions of the kidney are performed by these nephrons. Nephron is the unit, basic unit that perform urine formation and its concentration. Okay. A nephron is consisting of two parts. What are they? A nephron consists of two parts, namely the glomerulus. What is glomerulus? It is a tuft of capillaries. You are having a tuft of capillaries, and the renal tubule is the second part. So it consists of two parts. What is the glomerulus? Glomerulus is a highly coiled tuft of a knot of capillaries this is the glomerulus tuft of capillaries, and the second one is the renal tubule. What is that? It is a highly coiled thin walled tube which is having only a length of about 3 centimeter. It's a highly microscopic structure having a length of about 3 centimeter average. Okay. This renal tubule, second one is the renal tubule. Okay. So these are the two parts. This renal tubule has three regions. What are they? The first part is the cup-like part, a double-walled cup-like structure in the form of a capsule. So it's called Bowman's capsule. Bowman's capsule. This Bowman's capsule is a double-layered cup-like structure. It is having an inner layer, inner wall, and an outer layer or wall, which is made up of epithelial cells. Inner layer is made up of thin, flat, squamous epithelium. Squamous epithelial cells. So it's a thin, flat. And this cup will be enclosing the knot of blood vessels called glomerulus. Bowman's capsule plus the glomerulus it carries. Bowman's capsule plus glomerulus is called the Malpighian body or renal capsule or corpuscle. Renal corpuscle or Malpighian body. Renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus along with the Bowman's capsule is called Malpighian body or renal corpuscle. Is that point clear? So that is the first part. The second part is the tubule. The remaining part is the tubule, which is a highly coiled tube. Tubule is divisible into three regions. What are they? One, it is divisible into three parts, which are all the first part is a highly coiled part, so it's called proximal convoluted tubule. Proximal means very close to. That's the meaning. Close to the Malpighian body or renal corpuscle. So it's called proximal convoluted. Convoluted means highly coiled. You are having another highly coiled, which is the farthest away from the Malpighian body. So what can be the name? It can be given distal convoluted tubule. It is called DCT. These convoluted tubules are coiled tubes. That's all. I have drawn in an extended form so that its structure will be very clear to you. Okay. Actually, it is remaining in the form of a highly coiled tube. And these two tubes are interconnected by, or they are all connected together by a special structure which is the form of a loop or in the form of a U-shaped structure and is called Henle's loop. This Henle's loop loop of Henle or Henle's loop is a hairpin bend, just like a U-shape, hairpin shape part, and the two limbs of the hairpin, they are called descending limb coming down. That is the meaning. First part that follows the PCT is the descending limb, and the second part which is joining the DCT is called ascending limb, climbing, going ascending limb. Okay, so you are having a descending limb and an ascending limb for Henle's loop. Both descending and ascending limb are having a thick and a thin. This is the thin and this is the thick. Meaning a part which is having more diameter and a narrow diameter. Again, the thin limb in the descending limb is longer, whereas the ascending limb, it is the thick limb that is longer. In the descending, thick limb is shorter and thin limb is longer, whereas it is just the reverse. Thin limb in the ascending limb is shorter and thick limb is longer. Clear? Understand the differences between the descending and ascending limb of Henle's loop? So you are having a PCT and a DCT connected together by a hairpin bend like structure called loop of Henle. So this is the renal tubule. So it is having a glomerulus and a renal tubule. Renal tubule is having PCT, DCT and a Henle's loop. Clear? Now these renal tubules are all joining a straight tube. That straight tube is called collecting tubule. It is called collecting tubule. Why it is called a straight tubule? Because it is not at all coiled. It will receive several such tubules. So several urinary tubules join a CT, a collecting tubule, and several such collecting tubules join another tubule called duct of Bellini. Here I told you pyramids are having renal columns or column of Bellini. Here it is called, the duct is called Bertini. Don't get confused. Ducts of Bertini, that is not in your books. So CT is open to ducts of Bertini, and ducts of Bertini open to the surface of the pyramids. They are all opening to the surface of these pyramids. And urine is brought to the pelvis through them. Now urine is dropped into the pelvis and pelvis open to the ureter and urine is thus taken away through the two ureters to the urinary bladder. Remember, you are having about 1,000,000 such tiny, highly coiled tubules called nephrons, okay, which are the structural and functional units of each kidney. Any doubt about its structure? Anybody having a doubt? Okay. Now, this kidney is getting about 1/5 of the blood pumped by the ventricle. In the circulation, I have told you that each ventricle is pumping about 5 liters of blood per minute. That is the stroke volume into frequency. That is called cardiac output. It is almost 5 liters per minute. 1/5 of that is about 1 liter. So each kidney is receiving about 1100 to 1200 ml of blood per minute. Who is bringing blood? Blood is brought by the renal artery. So the renal artery brings blood at the rate of about 1100 to 1200 ml per minute, which is about 1/5 of the cardiac output. So kidney is receiving 1/5 of the blood pumped up by the ventricle per minute. And after entering into the kidney, the renal artery divides into a large number of branches called arterioles. These arterioles again branch into very thin branches which are called afferent arterioles. Afferent means bringing to. So these are all afferent arterioles, means bringing blood to the glomerulus. Thus blood is brought to the glomeruli through the branch called afferent arteriole. It brings blood. And after circulating the glomerulus, blood is taken out through efferent arterioles. So, from glomerulus, blood is taken by efferent. Efferent means taking away. Efferent arterioles. So, afferent brings blood to the glomerulus, efferent takes away. Okay. Now, this efferent arteriole, after coming out of the Bowman's capsule, again branches. It branches. This blood vessel again become it is branching and gives supply to the entire tubule, including PCT, DCT, Henle's loop, etc., etc. So it is giving large number of capillaries that surround the entire tubule. So we can call them as peritubular capillary. What's the name? Peritubular, meaning peri means surrounding, around. So peritubular capillary means capillaries around the tubule. So efferent arterioles are giving rise to peritubular capillary. And these peritubular capillaries are again joining to form veins. Capillaries join to form veins. Veins arise from capillaries. Arteries end up in capillaries. Veins begin in capillaries. In the last chapter, we have discussed it. Okay. So these veins will join. These are small veins. Okay. These small veins join to form renal vein, which is the main vein that takes away blood. This renal vein joins the inferior vena cava, taking blood back to the heart. So this is the flow of blood inside the kidney. So kidney is having abundant blood supply. It is receiving about 1/5 of the blood pumped by the ventricle. And the flow is, please understand, renal artery, arterioles, branches into afferent arterioles, give supply to the glomerulus, blood is taken from glomerulus by efferent arteriole and it forms peritubular capillary after coming out of the glomerulus, after coming out of the Bowman's capsule. Peritubular capillaries will form small veins which join to form renal vein that takes blood away from kidney. So this is the route of blood flow. Any doubt? Anybody? Anybody, Makla? Should I explain in Malayalam also? Is there anybody who cannot understand Malayalam? Please respond. If there is anybody who cannot understand Malayalam, okay. Now you are getting an idea regarding the structure of a kidney. Then there are two types of nephrons in our kidneys. What are they? They are called cortical nephrons, and the other one is called juxtamedullary nephrons. I will tell you what is the meaning. Cortical nephrons are called so because they are lying in cortex. That is the Bowman's capsule, PCT, DCT, and even loop of Henle are in cortex. They are small, okay, but majority of them, 80 to 85% of our nephrons are of this type. Since they are lying mainly in cortical region, they are called cortical nephrons. Majority of our nephrons are of this type. Here, loop of Henle is small. It is not lengthy and is not going deep into the medulla. മെഡുല്ലയുടെ ഉള്ളിലേക്ക് ആഴത്തിലേക്ക് പോകുന്നില്ല. It is also mainly lying in the cortex. Okay. Then juxtamedullary nephrons. What is the meaning of juxtamedullary? Juxta means lying closely, close to the medulla. So, for example, the cortical nephrons are lying here. Okay, all their parts are in the cortex, whereas in juxtamedullary we are having cortex only, Bowman's capsule, PCT and DCT alone, whereas loop of Henle is deep into medulla, going deep into medulla. Bowman's capsule, PCT, DCT are lying very close to the medulla. See, it is very close to the medulla. That's why it is called juxtamedullary, very close to the medulla. And here the nephrons are only 15%, very small in number, only 15% of our nephrons are of this category. Here, loop of Henle is long and go deep into medulla. Loop of Henle is very long and goes deep into the medulla. Okay. Then there is one more major difference between these two. What is that? I told you that we are having a peritubular capillary that is surrounding the tubule. We are having a peritubular capillary, and there is a special capillary which is a part of the peritubular capillary running very close to the Henle's loop. It is originating from peritubular and ending up in peritubular. So, it is also in the form of a loop, lying very close to the Henle's loop. Remember, Henle's loop is very long, going deep into the medulla. So, this blood vessel capillary is also going deep into the medulla, running parallelly, and in them, flow of blood is opposite to the direction of the flow of urine in Henle's loop. We will be discussing that later on. This special capillary is called vasa recta. What's that? Vasa recta. In vasa recta, blood is flowing in a direction opposite to the direction of the flow of urine in Henle. This vasa recta is well developed. Vasa recta is well developed. In juxtamedullary nephron, whereas in cortical it is poorly developed or even absent. It is even absent. Now you understand the differences between these two? So we are having two types of nephrons in our kidneys. Majority of them are cortical nephrons, which are smaller, lying entirely on cortex. Any doubt up to this level? Okay. So with that, we are completing the structure of kidney. Any doubt? Any doubt? Okay. Please respond. If you are having any doubt to be clarified, you can ask me. Simply type that point. Now, collecting ducts are all joining to form ducts of Bertini. Ducts of any doubt? 1100 to 1200, maximum 1200 of ml of blood is filtered by kidney per minute. Okay, which is roughly 1/5 of the blood pumped by the ventricle. Okay, now how urine is formed? That is the functioning of the kidney. After studying the structure, we will be going to the study of functions of the kidney. Main function, there are so many functions performed by the kidney. Main function is formation of urine and its concentration. It is not only making urine. What is urine? Urine is a solution that contains urea. Okay, it is only a solution that contains urea. That urea is not produced by the kidney, but urea is produced by our liver by a complicated procedure, a cyclical event called ornithine cycle. It is by ornithine cycle that urea is produced. Where? In liver. Urea is highly soluble in water. So the urea produced by the liver is getting dissolved in the blood, in the water of the blood, and through the blood it comes out of the liver, and from liver it is carried to the heart first, and from heart it is taken to the kidneys. Through the renal artery it is reaching the kidney. Okay, so liver produces urea. And what is the blood vessel that is taking blood from liver? It is the hepatic vein. So hepatic vein take blood from liver to the heart, and through that it reaches the heart. Hepatic vein joins the inferior vena cava, and from heart it is taken through the renal artery to the kidney. Now kidney simply filters that urea from blood. That is the function. And then concentrates. After filtration of this nitrogenous waste material from blood, the kidney also concentrates and forms a concentrated solution. Okay, let us see how these are all done. There are three important events. What are they? First one is the filtration process. That filtration process is called ultrafiltration. That process is called ultrafiltration. Which is also called glomerular filtration, because it is from the glomerulus that filtering is occurring. Blood is filtered at the glomerular level, so it's called glomerular filtration or ultrafiltration. How it is occurring? Let us see. What is the nature of this glomerulus and the Bowman's capsule in which they are all present? Let us see. As blood is passing through the glomerulus, filtration is occurring. It is the first process, number one. And why filtration is occurring? There are two conditions. One, blood is flowing through the glomerulus under high pressure, because glomerulus is a small capillary. Blood is passing through that under high pressure. So the pressure inside the glomerulus is double than that of the blood pressure present in any other capillary. And what is the nature of a capillary? We know that capillaries are having only thin-walled endothelium. We have studied the structure of artery, vein and capillary. It is having a thin endothelium, inner epithelium, squamous epithelium called endothelium. This endothelium is having a special feature. What is that? It is having some gaps between the cells. This endothelium is having some gaps which will be giving its permeability more. So, the permeability, flow of material across this cell layer, endothelium, will be about 100 times more. Endothelium is having 100 times more permeability. Remember, the pressure is 60 mm of mercury. So it is under high pressure. Second one, it is having high permeability. Why? Because there are some gaps called fenestra. This word is not in your books. So, as blood is flowing, you will be having some gaps called fenestra through which blood can leak. Okay? Second one, you are having the inner lining of the Bowman's capsule. So, the wall of the capillaries are having fenestra, and the inner layer, which is made up of squamous epithelium, is having a special type of cell, which is almost like the shape of the foot. So are called podocytes. These cells are having the shape of our foot. So it's called podocytes. Podocytes are present in the inner layer of Bowman's capsule. That inner layer is also called visceral layer. Visceral layer of Bowman's capsule. What is the peculiarity? The cells are having foot-like shape, so that even if the cells are kept close together, that is the characteristic feature of epithelium, they leave between them some gaps. These gaps are called filtration slits or slit pores. These two layers of epithelium are connected together by a thin layer of gelatinous material, jelly-like material that connects. Okay, so that is the thin layer of matrix. So, though there are, this is the matrix, 3 layers. What are the 3 layers? 1, endothelium. 2, matrix. 3, inner layer of Bowman's capsule. Because of the presence of slit pores or filtration slits, almost everything in the plasma, liquid part of the blood, is filtered out. So, the 2 factors that help in this filtration is called, or filtration are 1, pressure, 2, the presence of pores. So, the capillaries are, or the inner wall of the Bowman's capsule plus wall of the glomerulus together act as the filter. This is the filter that filters. See, when we are all making tea, first of all, we will be pouring the tea dust to a filter. Then we add water. Only the soluble part will be coming out through the filter. The large-sized tea leaf are never coming. Exactly like that. As blood is flowing, the liquid part of the plasma, that is plasma minus proteins. No blood vessels. Blood vessels are larger, so they will be never coming. Only the liquid part, that is called plasma, minus proteins will be coming out. So almost everything of the plasma is filtered. That is why this filtering is called ultrafiltration, complete filtration. almost everything except large size proteins are filtered. So that filtrate that is coming out will be first accumulating where they are all collected and stored in the capsular space. This is the space between the outer layer and inner layer of Bowman's capsule, this capsular space. This is called capsular space. To that liquid is collected and stored. That liquid is called glomerular filtrate or primary urine. First formed urine is primary urine. It is having the same concentration of minerals and salts as that of the blood. So it is said to be isotonic, having the same salt concentration as that of the blood. It is called ultrafiltration because complete of the plasma that contain essential materials that are required by the body, non-essential materials that are to be sent out of the body, everything. So, ultrafiltration is not a discriminated or a selective one. So, the first process of urine formation is called ultrafiltration or glomerular filtration, which is a non-selective. Why it is called non-selective? Because there is no selection. Even essential things needed by your body is coming out. Okay, that urine is called primary urine. From this primary urine that is accumulated in the capsular space, which is isotonic, it is slowly passing through the renal tubule. So first it passes through the PCT, then Henle's and then DCT. Is that point clear up to that level? Now let us see what is the second event. Remember, kidney is filtering 1100 to 1200 ml of blood. It is filtering 1100 to 1200 ml. And out of that, 125 ml of urine is filtered per minute. 125 ml of urine is formed out of 1100 to 1200 ml of blood. So almost 10% of the blood is filtered and coming out. Okay. This primary urine now passes through the PCT, then Henle's loop and DCT. Now, what is the second event? Let us see. The second event is called, first important event is the ultrafiltration. Second event is called tubular reabsorption. What is tubular absorption? As the urine is passing through the PCT and DCT, the walls are capable of absorbing back. They are all capable of absorbing back materials that are all needed to the body. Why it is reabsorbing? Because these are all materials that are needed by our body. How they are all absorbed? This absorption, understand it clearly, this absorption is taking place because the walls are having cuboidal cells. The walls are having cuboidal cells, meaning cells which are all cubical in shape, which are having abundant mitochondria and large number of microvilli. So they are having what is called a brush border epithelium. The cells are having large number of villi-like structures. That is why they are called microvilli. Both in PCT and DCT, the cells are cuboidal epithelium. Squamous epithelium is present in the Bowman's capsule, especially the inner layer. Okay. In PCT and DCT, you are having cuboidal cells, and as urine is passing, from that, materials that are needed by our body are absorbed. See, for our convenience, I am removing this. Remember, you are having the blood capillaries. What is the name? Peritubular capillary. To that, the materials are absorbed back. How? They are absorbed in two techniques. One, those which are all absolutely essential, like amino acids, glucose, sodium, etc. are absorbed actively. Thus, 70 to 80% of the electrolytes from that primary urine is absorbed back. From the primary urine, they are absorbed to the peritubular capillary. For that, energy is required. Yes, it is actively absorbed. Active absorption is energy requiring. So, complete glucose, complete amino acids are all absorbed. Now, there will be also absorption of other minerals like sodium. Along with that, there is some passive absorption of small amount of urea. This is passive. Okay, so 70 to 80% of electrolytes are absorbed as it is passing through the PCT. Similarly, as this urine is passing through the DCT, large amount of ions like sodium, then water, bicarbonates are absorbed. But the absorption takes place in the DCT is conditional. What is the meaning of that term? Only if they are all needed to your body, they will be absorbed. Otherwise, they will not be absorbed. So, absorption taking place in the DCT is conditional. What are they? Sodium. Okay. To maintain the sodium potassium balance, if sodium is required, it will be absorbed. Similarly, water. If your body is deficient in water, if the water is to be retained in the body, it will be absorbed. Similarly, bicarbonates to maintain the pH. Remember, our blood is having a constant and steady pH. It varies from 7.2 to 7.4. That is maintained by two salts. Okay, two. Bicarbonate is one. Okay, so sodium, but the water and bicarbonates are absorbed. That is conditional. So this is conditional. It is selective. Depending upon the requirement of the body, these materials are absorbed. So the second event is called, second most important one, is the tubular reabsorption. Tubular reabsorption is selective. It is selective. It takes place both in PCT as well as in DCT. Okay. So, essential materials are absorbed, and to maintain the normalcy, all such materials are absorbed. And third and the last one is tubular secretion. What is tubular secretion? See, when urine is passing through PCT and DCT, the cuboidal epithelium, which are all glandular, can also secrete from the surrounding blood. See, they are all surrounded by the blood vessels, peritubular capillaries. From that blood, they can absorb materials that are to be sent out of the body, but which will not be passing through the filter. These materials are not filtered through the pores. So, such materials that are to be sent out of the body, but which are not passing through the filter, are definitely separated from blood and pushed into, they are all pushed into the tubules as urine is passing through them. So what are such materials? Let me see. It include from the surrounding blood vessels, materials like hippuric acid, creatinine, etc. Also antibiotics like penicillin. Suppose you are all taking antibiotics, after their use, they are all to be sent out. So antibiotics like penicillin are all secreted from the surrounding blood vessels. It is taken and pushed into here. So hippuric acid, creatinine, antibiotics, etc. Here also, in DCT also, you are having the same type of phenomena occurring. And what are the materials that are all secreted here? You are having potassium ions. So sodium absorbed, potassium release. So that sodium potassium balance, equilibrium in the body is maintained. Here you are having hydrogen ions. So hydrogen and bicarbonates, positive and negative charge ions. That is how our body ionic balance and pH balance are maintained. Okay. So you will be having potassium ions, hydrogen ions, and ammonia secreted into. Thus, materials that are all to be sent out of the body but cannot pass through this filter are all secreted by the cuboidal cells. Remember, cuboidal cells are having large number of microvilli and they are having large amount of mitochondria. So, using that energy, these materials are sent into the urine as the urine is passing through the PCT and DCT. This sending out, secreting into the urine from the surrounding peritubular capillary is called tubular secretion. This tubular secretion is with the help of the cuboidal cells, both in PCT and DCT. Remember, this activity is the third and final action, which is also tubular secretion is also a selective one. So, you are having three important events in urine formation. Ultrafiltration is number one, which is the only non-selective process. Second one is tubular reabsorption. It is a selective process, and tubular secretion is also a selective process. So, it is with the help of these three activities that urine is filtered and brought to the CT. Is the point clear? So, urine formation is as a result of these three, namely glomerular filtration, reabsorption and secretion. Any doubt up to this level? You are having this diagram. So, bicarbonate, sodium chloride, water, etc. are absorbed here, and here potassium is not absorbed. Potassium is actually secreted into. Okay. So, you are having bicarbonates absorbed back, potassium, ammonia, small amount of ammonia and hydrogen ions. So, sodium chloride is absorbed, hydrogen is water is also absorbed. So, we are having tubular absorption as well as tubular secretion taking place both in PCT and DCT. So that the urine now will be entering into the collecting duct. Okay, collecting duct. But we have not studied the role of the Henle's loop. Role of Henle's loop will be discussed on the next class in detail. So, what is the role of PCT? PCT is playing an important role in absorbing materials that are all needed by your body and secreting materials into that. So, selective absorption and selective secretion, meaning it is helping in maintaining a constant and steady water and ionic concentration in our body. That is mainly the function. Okay, and we are having PCT is the site where 70 to 80% of the electrolytes and water are absorbed by this segment, and it plays an important role in maintaining the pH, acid-base balance and ionic balance of the body fluids by selective secretion of hydrogen ions and ammonia into the filtrate. So, secretion of hydrogen ions and ammonia into the filtrate and by absorption of bicarbonate from them. Okay, what is the role of Henle's loop? I will tell you later on. DCT, it is conditional reabsorption. Conditional means depending upon the requirement of the body. Sodium and water is conditionally absorbed in this segment. It is also capable of reabsorption of bicarbonate and selective secretion of hydrogen and potassium ions and ammonia. But you understand that? I have marked it that ammonia also in your book it is not given. To maintain the pH, that is the acid-base balance, and sodium potassium, that is salt balance. So, maintenance of acid-base balance and sodium potassium, that is salt balance, that is ionic regulation. That is the function of DCT and PCT. Okay. Now, what is the role of Henley's loop? Henley's loop is not playing any much role in this maintenance of osmolarity. Okay. But it plays a very important role. Henley's loop, here reabsorption is minimum and it is not playing much role in maintaining the salt water balance, etc., but it is having some important functions which is helping in maintaining a high osmolarity gradients. What is that? We will discuss next class. So, do anybody have a doubt on this part? Okay. So, it is time for me to wind up my session because the time is over and we will continue our discussion next week. Until then, goodbye and good night. So, thank you all. Thank you. Thank you.